Hello, today I'm going to be presenting my research investigating the neuroprotective effects of astaxanthin on mobility, protein aggregation, and lifespan in C. elegans amyotrophic lateral sclerosis models. To start off with some background, amyotrophic lateral sclerosis, also known as Lou Gehrig's disease, is a fatal neurodegenerative condition that primarily affects motor neurons, leading to death typically within 2 to 5 years of diagnosis due to a lack of effective therapies. ALS involves high levels of oxidative stress in motor neurons. This stress results from electron leakage on both sides of the inner mitochondrial membrane, associated with abnormal protein aggregates from the human TDP43 gene, which is crucial for RNA and mRNA processing under normal conditions. In ALS, however, these aggregates contribute to the loss of motor neuron function, causing muscle weakening, accelerated disease progression, and ultimately death. With over 300,000 individuals affected in the United States alone and no cure available, there's a substantial unmet need for treatments that alleviate ALS symptoms. To address this, I modeled ALS in C. elegans, seeing as they boast a well-mapped nervous system to replicate human ALS. The specific mutation in my study enhanced TDP43 aggregates in the mutant C. elegans, mimicking ALS pathology. During my research for potential treatments, I identified astaxanthin, a natural antioxidant derived from seaweed known for reducing oxidative stress and inflammation. While astaxanthin has shown potential in alleviating symptoms in other neurodegenerative models like Alzheimer's and Parkinson's disease, Baba et al. 2021, its effects have not been tested in ALS-specific C. elegans models. My study aimed to fill this research gap by exploring astaxanthin's effectiveness in mitigating ALS-related symptoms in these nematodes. My research question for the study was, how effective is astaxanthin in reducing protein aggregation and improving motor function in TDP43 mutant C. elegans models of ALS? As for my hypothesis, I hypothesized that astaxanthin supplementation would reduce protein aggregation, improve motor function, and prolong lifespan in TDP43 mutant C. elegans compared to untreated mutants and wild-type controls. As for my methodologies, my control groups were wild-type C. elegans and untreated TDP43 mutated C. elegans. These mutated C. elegans displayed a phenotype that showed characteristics of lower mobility rate, lifespan, and reactive burrowing when exposed to oxygen. As for my experimental groups, I exposed the mutant and wild-type groups to different concentrations, 10, 50, 100, and 200 micromolars of astaxanthin, fed directly to the nematodes orally. The dependent variables I am measuring included motor function, lifespan, and protein aggregation. First of all, the thrashing assay was used to evaluate neuromuscular function in C. elegans, with the number of thrashes the nematodes did per minute counting as a quantifiable measurement of motor neuron function. The thrashing assay shows that while the untreated mutant type showed a significantly lower amount of thrashes than the wild type, astaxanthin treatment led to a dose-dependent improvement in thrashing rates, with the highest doses restoring mobility close to wild type levels. This suggests that astaxanthin can alleviate neuromuscular dysfunction caused by TDP43 aggregation. Fluorescent microscopy was then used to visualize and analyze TDP43 protein aggregation in the neurons of C. elegans. Mutant worms expressing GFP-tagged TDP43 proteins were imaged using a fluorescent microscope, while untreated mutant C. elegans displayed high levels of TDP43 aggregation along the spinal areas. Astaxanthin significantly reduced visible aggregation in a dose-dependent manner. Higher doses showed minimal aggregation, supporting its efficacy in reducing protein misfolding. Finally, the lifespan assay was conducted to assess the effects of astaxanthin on the longevity of TDP43 mutant C. elegans, treated and untreated. Lifespan analysis revealed that astaxanthin treatment significantly extended lifespan in a dose-dependent manner, demonstrating a protective effect against the toxic consequences of TDP43 aggregation. Kaplan-Meier survivor curves show clear dose-dependent trends, supporting astaxanthin's therapeutic potential in ALS models. In conclusion, this study yielded remarkable results supporting the hypothesis that astaxanthin has promising therapeutic effects in TDP43 mutant C. elegans, mitigating symptoms of ALS. Higher doses, particularly 200 micromolar dosage of astaxanthin, significantly improved mutant C. elegans mobility to near wild-type levels, reduced TDP43 protein aggregates in neurons of mutated C. elegans, and extended lifespan of mutant C. elegans in a dose-dependent manner. These findings highlight astaxanthin's potential to mitigate neuromuscular dysfunction and protein misfolding in ALS models. Based on the results of this study, astaxanthin shows great potential for future applications in improving the quality of life of many people. Astaxanthin's remarkable ability to reduce TDP43 aggregation in C. elegans suggests potential applications for mitigating the devastating symptoms of ALS and similar neurodegenerative disorders. The study also showed the therapeutic role of antioxidants in maintaining proper protein synthesis, which can be explored for other protein aggregation diseases like Huntington's and Parkinson's. As for future research, I believe the best place to start would be to optimize the dosage of astaxanthin administered to C. elegans, because as there was a dose-dependent reaction to astaxanthin administration in my study, I would like to investigate the optimal concentration of astaxanthin for maximum therapeutic benefit with minimal toxicity. Additionally, the mechanisms by which astaxanthin actually reduces oxidative stress is not yet known. Through this research, we could potentially apply astaxanthin treatment to other disease models. Thank you!